Good evening, everyone. Welcome to Season 5 of Lightning Talks. We are with you live from the Seattle Aquarium on the shores of Puget Sound on the traditional and contemporary territories of the Coast Salish people. And we are thrilled that you're joining us tonight from wherever you may be this evening. My name is Nora Nickham. I use she, her pronouns, and I'm the Vice President of Conservation Programs and Partnerships here at the Seattle Aquarium. And tonight's Lightning Talks episode has the theme of spineless. Now, why spineless? Well, one reason is that we are here to talk all about invertebrates, and invertebrates are animals that do not have a spine. But we also thought it was a fun title, and I think one of the reasons is because it's actually a pretty big mismatch when you think about the second definition of spineless, which is weak or lacking in courage or spirit or determination. And I'm pretty sure at the end of this episode, after meeting all the creatures we're going to hear about tonight, we will all want to tell Merriam-Webster to revisit that second definition. So if you are new to Lightning Talks, let me quickly explain how it works. We have 5 speakers, and they each have 5 minutes and not 1 second more to share something that they're excited to share with you all this evening about spineless creatures. And as they go along, as they're getting low on time, they're going to get some warning lights. So when they have 1 minute left, they're going to see this. When they have 30 seconds left, they will see this orange dot. And then when they're almost out of time and then run out of time, this is what happens if they keep talking. And that's what gives Lightning Talks its name. So we always hope it happens at least once. Now, we hope that all throughout you will be hearing lots of things that make you really curious. So please put a question in the chat anytime. There's also a QR code you can use and a link. And so as things occur to you, please put them in there. We will be holding questions until the end, but then we're going to bring all of our speakers back for a great conversation, and we'd be pulling from a lot of the questions that you all pose. So before we get started, I wanted to invite anyone who enjoys the show tonight and would like to support the Seattle Aquarium in continuing our mission of inspiring conservation of the marine environment to consider a donation to help keep these events going. Now, without further ado, I would love to bring up our first speaker, Dr. Drew Harvell. Welcome, Drew. Drew is affiliate faculty in the University of Washington School of Aquatic and Fisheries Science. She's a former aquanaut with NOAA, and her diving research on the health of corals, seagrasses, and sea stars has taken her all over the world, from the reefs of Hawaii, Mexico, Indonesia, and Palau to Friday Harbor Labs right here in the Pacific Northwest. She's the author of a new book, which I highly recommend, called The Ocean's Menagerie, which is all about spineless creatures. And she's going to introduce us to some of those creatures now. Drew, are you ready to kick us off?
Ready to go. Hey, Nora.
Your slides look great. Go for it.
All right, ready to start. This is about the best topic ever. These are the 8 critters that star in my book, and I like to talk about the superpowers of the spineless, and I'll share a few vignettes of those tonight. My background is as a graduate student, a PhD student, we won't say how long ago, at the University of Washington. I got invited along to be an aquanaut with a research team. I subsequently did 4 missions in this Aquarius underwater lab at 60 feet, living underwater on a reef. So it just set my compass for my entire career to spend a lot of time diving underwater doing research. And so I learned a lot about bio-inspired design, and I'm going to share 3 vignettes: cancer drugs from sponges, transplantation biology from nudibranchs, and sea stars' sticky skin. So what about the sponges? Well, we did spend quite a few years in natural products chemistry cruises, and sponges were the top hit rate in finding new anti-cancer leads. And I'm showing here one of them, this swinoolide. It's kind of special. It has a lot of activity, but it turned out that it's actually made by bacteria living inside the sponge. We used to think the sponges made it all. It's turning out a little bit deeper that it's the bacteria. And that's not the only case. Helicondria panacea, I bet half the people here know it. It's really common in our tide pools. It houses a bacterium that makes miomycin active against 5 different carcinomas. A related species, Helicondria acadi, produces also bacteria produce another compound approved by the FDA. So I'm saying here the superpower of these sponge bacterial symbioses are making complicated chemistry or drugs. Now, this is one of my favorite nudibranchs. So this is the next vignette. This is Aeolidia in one of our tide pools on San Juan Island, and it hunts sea anemones and will eventually eat it. And it eats the whole thing, including the stinging cells that are in the sea anemone. And the superpower that I like to talk about of nudibranchs is what's called kleptonidae. Now, here's a bunch of our critters from Friday Harbor labs, typical lab prep, but only one of them actually makes the kleptonidae. And it's this guy in the upper right. I don't know if you can see my cursor. And it actually uptakes the stinging cells and has tricks for doing it. So how does kleptonidae work? The nudibranch uptakes the stinging cells, passes them all the way through their digestive system. Then these handy little projections on the back are actually digestive gland, and they house those stinging cells, and they're responsible for them maturing. So I talk about them as being a good idea for thinking about new transplantation methods. The sea star's sticky skin is my last vignette. And I just want to say how cool this is that at the touch of a neuron, these collagen fibers get cross-linked and the skin gets absolutely stiff. So our star can open a clam. And also at another neuron's touch, it dissolves. And so our star can autotomize its arm. And so I'm calling the superpower this sticky skin or mutable collagenous tissue. Now, there's other cool things going on with sea stars. We've been studying the outbreaks of wasting disease for over a decade, and we're into the sunflower star recovery plan. And our group has been working on the epidemiology of the disease and making some good progress in the causative agent and in studying the immune system of our sea stars. And so I want to conclude by saying tonight it's spineless in Seattle and invertebrates are it. Terrestrial life is a bit of a sideshow in the evolutionary drama. Whales are, of course, cool, but invertebrates run the gears of ocean life. And I hope you'll be able to come join me at some of my other book talks at Elliott Bay or Seattle Town Hall. And here are some of the other topics in my book, since I only could do 3, which I can't even believe I got through those 3. But there's things like the coral's castle, the octopus's shape shift, and the jellyfish's light show. And I think I got through before the lightning struck. So I'm going to be happy if there's any questions that any of you have later. And my timer says I'm just about done. So
you did a great job, Drew. Thank you. I will say that of the other chapters in the book, one of my favorites was the one about the giant clams light trick. So I do encourage people to go pick up a copy of the book and read about giant clams. But you know, something you said early in your presentation sparked my interest, and I'm sure sparked the interest of some of our viewers too. You said you lived underwater, 60 feet down, I think. Can you tell us a little bit more about what it's actually like to live down in the ocean?
Well, it was pretty incredible as a grad student to be invited along to be part of this research team. There were 4 of us that lived in the Aquarius habitat, and it was definitely the coolest thing probably I ever got to do. And the amazing thing was to just watch the day-night shifts. In the morning, we'd watch the reef fish swim off the reef, and in the evening, they'd come back in. And then at night, we would do night dives, and we could even find sleeping fish on the reef. And so it just was like being part of the reef. Pretty remarkable privilege to be able to do it. And we went back and did it 3 more times. So
amazing. And how do you find your way back home after a night dive?
Oh, such a good question. So yeah, because if anything happens, you're saturated, you cannot go to the surface or you will instantly get the bends and be in a very bad way. And so there are ropes all along that NOAA has strung on the bottom of the reef with little metal arrows. So in the event that your mask gets ripped off and you can't see anything, you can always follow the rope back and get back to your habitat.
Wow. So it's all set up for safety and adventure. Thank you, Drew.
You're welcome.
So if anyone else has questions for Drew, please go ahead and put them in the chat. And in the meantime, we're going to welcome our next speaker to the Lightning Talk stage, Sarah Brenkert. Sarah is a social science researcher and the principal evaluator here at the Seattle Aquarium. And she also teaches evaluation in the University of Washington's Master of Arts in Museology program. Sarah studies how empathy for animals relates to conservation action. So tonight she's going to share about building empathy for spineless creatures, including some that we all might be able to meet out in a tide pool or on the beach sometime. So Sarah, I'll turn it over to you. Your slides are great.
Thanks so much, Nora. I'm excited to be here. And I want to start with a question for all of us. Now, no judgment, but why is it that we find it so much easier to care about pandas than plankton? Well, it's not that invertebrates aren't important. They do literally everything. They pollinate, they decompose and build soil, and they sustain entire ecosystems. But while invertebrates are everywhere, they get just a tiny fraction of our love and concern. Now, it's not really that surprising. Most conservation efforts have focused on the charismatic megafauna, right? Those big photogenic animals that we just naturally are gravitating towards, like pandas and elephants. Basically, if it could star in a Disney movie, it's charismatic megafauna. And at the Seattle Aquarium, we get lots of oohs and ahs about our sea otters and our northern fur seals, those big rizzy mammals. But because invertebrates are so important to marine conservation, we were curious if we could build connection and even empathy for them. The animals that seem strange or weird, the animals without faces, the animals that people aren't even sure are animals. So we set out to understand how we could do this. And because we're all giant nerds, we started with the science, specifically the research of Dr. Eugene Myers. And his work helps us understand the science behind why people generally find it so much easier to relate to pandas and polar bears than to sea stars and coral. It turns out that much of it comes down to anthropomorphism, which is basically our tendency to put humanness at the center of everything. Dr. Myers identified 4 characteristics that predict how easy it is for people to relate to an animal. And spoiler alert, they all echo characteristics we see in ourselves. The first is agency. Agency helps us connect to an animal because it's when we see them trying to do something, to make a choice, to solve a problem. That helps us see the animal as an individual with needs and goals. The second characteristic is affectivity. This is when we perceive, or, okay, let's be honest, project feelings or a state of mind on an animal. They're bored, they're lonely, they're excited. Now, we're not particularly good at this. We tend to do it in human terms. Once again, anthropocentrism. But it turns out that it actually doesn't matter all that much if we're accurate in assessing how an animal might be feeling. It has more to do in whether or not we believe that animal can even have a perspective or a lived experience. A third characteristic is continuity. This is just familiarity and time spent with an animal. Pets, backyard birds, the animals that we see in popular media. It's why we feel like we know them. And finally, the fourth characteristic is coherence. This one's interesting. It's whether or not something even registers as an animal in our brains. So the way that the human brain constructs knowledge is by a series of progressive remodeling of cognitive schema. We all have rules about how the world works and things fit into different buckets. And this is true for adults and young children. So, for example, you might have never heard of a pangolin, but if I were to show you a picture, you'd probably be pretty confident in being like, yep, legs, tail, face, that checks, right? Definitely an animal. However, when we look at something like an anemone, it doesn't necessarily check all of our animal boxes. And when we have trouble wrapping our heads around the coherence of an animal, it often means we don't relate to it as well. So why does this matter? Well, 90% of marine species are invertebrates. Invertebrates are the foundation of ecosystems, and it might be that finding empathy for these invertebrates is crucial to our own survival and the health of our planet. There's some promising research that suggests that if we want to change how people act, we have to start by changing how they feel. So how do we do that? Well, take the anemone here. Both the animals in these pictures have arms, eyes, and a mouth. They're just arranged a little differently. For the anemone, the arms are in a circle around its mouth. The eyes are light sensors at the tips of those arms. And they're sensitive. They respond to touch. If you put your finger out there, those arms will curl around it, sensing friend, threat, food. And when we think about an animal in those terms, we can start to see its agency. We recognize its coherence, and maybe it becomes a little less strange, a little more alive. The way we talk about invertebrates can help to close the psychological distance that we might feel. We can practice framing them using those characteristics, agency, affectivity, and coherence. We can tell stories about individual animals, and we can practice critical anthropomorphism, where we stay centered in scientific accuracy, but we look for parallels and similarities for how animals meet their needs, make their homes, and explore their world. And with that, I think I'm at time.
You are wonderful. Thank you, Sarah. It's amazing. And I just think it's wild that there are so many animals out there that we can't necessarily tell at first glance are even animals. That's so interesting. And then you can still build an emotional connection if you kind of know a question to ask or a way to think about it.
Absolutely.
So at the end, you use the term critical anthropomorphism. And I'm just wondering if you can talk a little bit more about that and how that might differ from other kinds of anthropomorphism, or maybe an example.
Yeah, absolutely. And I think for a lot of us who have worked in animal care or biology, any kind of science-based field, anthropomorphism has been kind of a taboo idea because we associate it with projection, with that anthropocentrism of projecting human emotions or human traits onto other animals. But critical anthropomorphism is actually emerging a completely distinct construct, and it has to do with staying really grounded in the scientific integrity and the biology of an animal, but seeking to draw parallels between how an animal survives, how they find shelter, how they find food. So that example that I gave of just talking about the anemone's arms and talking about how it might be sensing whether you're a friend or a threat. Do anemones really think about friends? We don't know, right? We can't know. But when we put it into terms like that, it can help to sort of be a bridge to make those animals more relatable because we can see parallels to our own existence and our own lives.
Wonderful. Well, later in the conversation, I hope there'll be a chance to ask you about how we might think about some of the animals we're going to hear about next.
Yeah, definitely.
Look forward to that chat. So everyone, please put some questions for Sarah in the chat. And we're going to introduce our next speaker, Kate Stanley. Kate is a senior aquarist, and she cares for the fish and invertebrates that visitors see here at the Seattle Aquarium, the ones that are local to our waters here. And Kate is particularly interested in figuring out how to care specifically for invertebrates that are local to the Salish Sea. You ready, Kate?
I sure am.
All right, your slides look great. Go ahead.
Thanks, Nora. So of course, we all talked about how we're nerds. One of my favorite ones is crabs, and I wanted to kind of share the species that my team are kind of the favorite ones that we keep in our collection here. We're all used to seeing crabs like this. This is typically how most people interact with crabs, but I'm going to share some fun facts and then our kind of some favorite species we have. So crabs evolved around 150 million years ago, which means they were around during the Jurassic area, which means they kind of watched some people's favorite dinosaurs come and go from the face of the earth. Crabs are put into two groups, true crabs and false crabs. False crabs are what people normally and generally think of as hermit crabs. It's believed that some crabs can detect magnetic fields. You can see that in some crab traps. They'll have kind of types of metals that sometimes the idea is they take the charge off those traps so the crabs don't kind of run away. You also see that in the Christmas Island crabs. People think that they use the magnetic fields to find their way back to the ocean when they need to go reproduce. Crabs are also kind of the only group that can live almost anywhere on the planet as well, which is my favorite fact about them. There are crabs that actually live the majority of their life in trees, which means they're mostly terrestrial. You have crabs that live in fresh water. We even have crabs like the Yeti crab that lives down in the depths by hydrothermal vents that are down on the ocean floor. Crabs can also regenerate limbs when they molt. You'll slowly see the different parts of their body that are segmented, which allows them to do that. And when a limb is lost, it doesn't come back to the normal size. It takes a few molts and a few cycles before it's back to that normal size as the rest of its pieces of its body. There are also a wide variety of sizes. The smallest crab is 5 millimeters. The largest crab has a leg length of up to 12 feet, which I think is pretty amazing. And crabs are really important in the ecosystem, from cleaning up decomposing materials, like Sarah mentioned earlier, to being food sources to a lot of variety of other animals. And so they kind of help clean up what other species would leave. Now I'm going to go on to some of our favorite species that we have in our local waters. So the northern kelp crab is one of my favorite species of crabs because of this shape of its carapace. It's kind of like a shield. So they are also unique in that their diet changes seasonally throughout the year. Their preferred diet is kelp, which they normally eat during the warmer months when it's growing and they're able to find it. In the winter months, when it goes away, they kind of switch over to being whatever they can find on the floor, and a lot of times it is decomposing materials from animals. They also have a wide range from Alaska down to Mexico on the West Coast, which means you can basically find them anywhere if you're living over on the West Coast. They also have the unique ability that if they're eating a lot of red algae or kelp that you can see on the top photo, they're going to be more of a red color. If they're eating a lot of maybe bull kelp, which is green in color, their carapace can change colors between molts, and it's really dependent on what is their current diet that they are eating. Next is the Puget Sound king crab, which is a favorite of a coworker of mine. These guys can get really big. Their carapace can be up to 12 inches wide. They also have a range of Alaska to California, which means you can find them in a lot of places. But over the years, they are getting very rare in the Puget Sound. So generally, you're finding them out straight to Juan de Fuca or on the coast where you have a lot of high flow for them. They like to eat different echinoderms. Echinoderms are stars or urchins that you would find. They also will eat almost anything they catch. And for us as caregivers of them, we kind of have to be careful and reduce the tank mates that they would have. These guys are also cool because their colors change. When they're younger, they have a lot more pink in their shells. And once they get to be older, it kind of goes more dull. The black-eyed hermit crab is not a true crab. It's one of the largest ones we have, of up to 2 inches. These are our favorite of our staff around here just because of their bright colors and their large eyes. Last one is the scaled crab. These guys are unique because of their pinchers are rounded and kind of shoveled. They prefer to eat brittle stars, which is also a type of type of echinoderm. And they have a symbiotic relationship with some plumose anemones, which you can see I found one hiding amongst plumose anemones in that top picture. And just like that, I'm all done.
Wonderful. Thank you, Kate. Well, Drew was talking about invertebrate superpowers, and I think you gave us some great examples of crab superpowers with the ability to grow 12-foot long legs for some of them, and regrow legs that they've lost, and follow magnetic fields. So it's pretty amazing. I was curious about the Puget Sound or the kelp crab. Do we have those here at the Seattle Aquarium?
We do. They tend to be housed by themselves, and so you can find them in some of our smaller habitats that we have around the aquarium.
Okay. And do you change their diet seasonally to reflect what they would be eating in the wild, even though you might have access to things year round?
It kind of just depends on what we have access to. We actually are kind of flipped. We have more access to kelp availability, not in the summertime, just because that is when our bulk kelp is growing in the underwater dome exhibit. And we harvest that bulk kelp, and that's actually what we feed out to our herbivores throughout the year. So for us, it's easier to give them bulk kelp in the wintertime than the summertime.
Wow, that's amazing that you harvest things here in the aquarium to feed some of the animals.
Yeah, it's one of my favorite parts of the underwater dome.
Oh, that's amazing. Well, I hope those who haven't come to see our underwater dome can come visit soon, and also look for that kelp crab in the smaller habitat. So please put questions for Kate in the chat. and we will look forward to welcoming you back, Kate, shortly for more discussion. And in the meantime, I'm going to bring our next speaker onto the Lightning Talk stage. So that is Luan Roberts. Luan is a marine naturalist and science communicator obsessed with sea slugs, dock fouling, and all things intertidal. She explores the Pacific coast, documenting nudibranchs in overlooked habitats and sharing her findings through zines, photography, and educational storytelling. And she has a great website we'll make sure is linked in the chat. So take it away, Luan. Make sure your slides are up. They look great. You're all ready to go.
Hi, I'm Luan, and I'm here to help you find 5 species of nudibranchs, also called sea slugs, that are living right here in Seattle. Nudibranchs are glowing weirdos, shapeshifters, hunters, and hermaphroditic icons of the sea. Finding one cracks open your awareness. You start noticing more, looking closer, and becoming part of the ecosystem instead of just walking past it. There are 2 main body types. Dorids have gills on their back ends, like frilly little butt halos. Aeolids have finger-like structures called serrata along their back, which help them breathe, digest, and sometimes even sting things. Nudibranchs live where the water touches the land, on docks, in tide pools, on old ropes, kelp, trash, rocks, pilings. If it's wet and crusty, it's worth checking. If you want to find nudibranchs, look for the eggs first. They often lay them before they crawl off to be invisible again. Each species lays a different kind of ribbon: spirals, ruffles, zigzags. When you see one, you're not just near a slug, you're in the middle of its story. This is a northern leopard dorid, yellow with peppery spots and a texture like lemon cake left out in the rain. It can get large, about the size of your hand. They blend in with sponges, so you'll miss them unless you've trained your eyes to spot the difference between lumpy and sluggy. Found in tide pools, docks, and sometimes right in front of your face. If you see one, you'll start seeing them everywhere. Introducing the white-lined dirona. The delicate frond-like projections are serrata. They help them breathe and digest. You'll mostly find them on docks, especially near broken posts, old nets, or shady overhangs where their food source hydroids grow. Check out how it glows against the dark. This one's under a dock hunting for prey. The light catches its serrata and makes it look bioluminescent, but that's just how the light reflects against its edges. This one's actually crawling on the surface tension of the water. A lot of nudibranchs can do this, so don't just look on rocks and ropes, look on the water itself. This one is a Monterey dorid, a large, firm-bodied dorid with a bumpy surface and bright yellow color when wet. It can grow to be a bit larger than your hand, and it tends to stay in exposed areas, such as the one spotted on the side of a dock. This one was on a dock piling in Seattle. They hide right out in the open. You'll often see them crawling in plain sight, even in bright light, usually on hard, bare surfaces. These 2 are out of the water, probably left high by the tide. If you find one like this, don't try and pry it up. Their bodies are tough but delicate. If they're already loose, it's okay to gently relocate them to a safer wet spot. This is a shagrug nudibranch. It's a big, fuzzy-looking aeolid that comes in all shades of gray and brown. It looks like the marine equivalent of a mystic lint trap, and it can be found everywhere. This one's crawling across a moon snail egg collar, one of their favorite hangouts. Shagrugs love sandy bottoms and silty docks, and they're often drawn to places where aggregating or plumose anemones are abundant. They eat sea anemones without even flinching. Their serrata, those frilly projections, help them digest and store the stingers for their own defenses. You'll find them on docks, often near the anemones on flat, bare panels. The final slug is Hermesenda crassicornis, the horned nudibranch. This is the Pacific Northwest classic. Has those electric serrata with the white stripe, glowing stripes along the side, and major main character energy. You can find them just about anywhere. This one's on a blade of eelgrass, but you can find them on kelp, ropes, dock posts, seaweed, and even on trash. This one's cruising across the moon snail egg collar. Yes, those weird jelly-like donut things that you find in the sand. Slugs love to hang out there, either laying their eggs or snacking on someone else's. If you look close, you can see those rhinophores, the tiny feathered antennae on the front of its head, and the glowing serrata tips. Every part of them is sensing, pulsing, tasting the world, often found right at the edge of the surface. You can find horned nudibranchs absolutely everywhere. I could keep on going forever on this slide, showing you the different locations that you can find them on an old bit of rope and some algae.
Yay for lightning. And that makes a great point, Luan, that there are so many places you could find these creatures that you cannot possibly stop within time limit. So wonderful. And I loved the tip about look anywhere wet and crusty. So that's great. I'm wondering if you can give us a few more tips for if we want to go out to a dock and try and find nudibranchs. What do we bring, or do we lie down on the dock and look underneath? Do we need a flashlight? When do we go? Anything that might increase our chance of success?
Absolutely. So this time of year is great because you have good light and the docks aren't too wet. So just having something to lay on, because there's a lot of seal poop on the docks and bird poop. So you want to get comfortable, but then you are just laying flat on the surface of the dock and looking over the edge. And where we have quite a bit of sunshine right now, it's better to look in the darker spots, because other spots might get like a bit too overgrown with algae and diatoms, and it can be hard to find things. But if you look like in a dim corner, then you have a good chance of finding a nudibranch, especially if there's something dangling off the side that you can examine a bit closer, like a piece of kelp or an old rope or a float.
So what's your success rate now, and has that changed over time? So are you pretty confident at any dock you can find a nudibranch if you know what to look for? And has it gotten easier, or is it still kind of a difficult treasure hunt?
It's definitely gotten easier. I think once you get the pattern in your head, and that's why I showed so many of the same images of the slug, is once you kind of get that pattern locked in, then you're able to like search for it a lot easier. So it definitely becomes easier. And there's always a sea slug on a dock. Like there might just be one, but I've never found zero. There's always evidence of them somewhere.
Okay, well, that's very encouraging for all of us to go out and try. So thank you. And so we have one final speaker for this evening, Brianne Ankenman. Bri is a senior aquarist on the temperate fish and invertebrate team here at the Seattle Aquarium, working with local species. And one of her many interests is learning new ways to improve animal care in aquariums by monitoring and evaluating environmental conditions and trying to match things to the specific native habitat of the animals we have. So she's going to be talking about an amazing animal called the basket star and how aquarists like herself work to make sure that their conditions are just right here at the aquarium.
Hello, everybody. So pictured here is the star of the show, pun intended, Gorgonocephalus eucynemius, also known as the basket star. A little fun fact for you, the genus name Gorgonocephalus is derived from gorgos, which is Greek for dreadful, and cephalus, which is Greek for head. So dreadful head is in reference to basket stars looking like the head of Gorgon in Greek mythology, like Medusa. The team here at Seattle have cared for basket stars for a couple decades and are always learning something new about their unique care. There is a lot to know about basket stars. I can only provide a small snapshot of their life history and care. So without further ado, here's a photo of a bunch of basket stars in their preferred habitat, which is a cold, dark cliff edge with strong variable currents. Basket stars are found in the Pacific Ocean from the Bering Sea to Southern California, parts of the Arctic Ocean, and the North Atlantic Ocean. Here in our local waters, basket stars tend to make their homes at depths ranging from approximately 50 feet to 500 feet. However, they can be found as deep as 6,200 feet. Basket stars are echinoderms, which we talked about earlier briefly. The larger group or phylum to which sea stars, urchins, feather stars, sand dollars, and sea cucumbers also belong. However, despite the name basket star, they are most closely related to brittle stars and are in a separate class from your typical sea star species. From a conservation perspective, the good news is that basket stars aren't listed as endangered or threatened. The less good news is that they can still be negatively impacted by fishing gear, seabed mining, and climate change. Their greatest threat is human activity. So that means it's up to us to vote to protect their critical deep water habitats. So a little bit about basket star form and function and their anatomy. The basket stars have 5 branched arms that are further branched into what are called branchlets. The overall shape of their arms creates a net or a basket-like structure, hence their name basket. Within those branchlets are tiny little hooks along their tube feet to allow them to ensnare and capture their prey. Now, the challenges in an open system aquarium, the cold water exhibits at Seattle Aquarium operate an open system in that the temperature of the water in the Salish Sea directly impacts the enclosure inside the aquarium. We were limited on where and how we could house the basket stars under our care initially. In years past, we learned to only house basket stars in tanks that have chiller systems to maintain colder temperatures, even into the hottest summer months. Otherwise, they would perish without that consistency and temperature control. Remember, these baskets like it nice and cold. Once we had temperature under control, we focused more closely on diet and feeding. Critical to basket star care is to offer food to the animal that is similar to what they would eat in the wild. Basket stars are reportedly seen feeding on plankton and small crustaceans floating in the currents. The optimal feeding behavior we want to see is the animal open like a basket. Then we want to see the animal capture the food particles in their arms, watch it curl, then bring the food into their mouth. Now, the food cannot be too big, otherwise it's too heavy to bring in. And if it's too small, they may have trouble getting enough food before it flows out the tank and down the drain. We have tried a variety of different food items in the past and currently offer a blended slurry of krill, small crustaceans, and other vitamins that are good for invertebrates. And here is a video. Strategies we have developed over time to feed them is to use essentially a turkey baster to target feed the animals with slightly larger pieces of food, as well as broadcast a blended slurry of food into the circulating flow of the tank, as seen here in this video. Changing the flow from a strong, fast current to a slower flow during feeding showed increased feeding behavior. We also have found that offering food to these animals multiple times a day leads to greater success in their overall well-being. We have used multiple strategies with the primary goal to allow the basket star to bring those branched arms down and into their mouth on the underside of that central disc. Other things we have tailored over time are lighting upgrades to simulate more of a natural light cycle with variable intensities of light throughout the day. We have also used different additives to maintain optimal water quality parameters or to simulate the alkalinity of their natural habitats. And in the last year, we have been implementing periodic immersion treatments with a probiotic. Our team became keen to this method from a recent study done at Oregon Coast Aquarium with sea star species. It has slowed down the presumed negative impact of pathogens in the water. In observation, this usually means the basket star has dropped branches and parts of their arms, so it's become one of the ways in which we can intervene in this stress response. It's critical to slow down the dropping of arms because with fewer arms, the animal will have a more difficult time feeding. They can grow back the branches of their arms, but it takes considerable time to heal. This last
Great. Finish up that last thought.
Yeah. It was just a video of a basket star climbing because that's what they like to do. And I just wanted to show one little video.
And how fast can they climb?
You know, my video was in real time and I'd say it's probably an inch per second.
Wow, that's faster than I would have guessed. So I have a question for you. I really like the photo that you showed of actually quite a few basket stars out in the ocean somewhere. And I'm wondering if you have ever had the chance to see a basket star in the wild.
I have, yes. So a group of our team will go out to Nia Bay every summer for collections for the aquarium. And one of the dive sites there is called Slant Rock. And so it's just actually a slanted rock, believe it or not. And basically, as a scuba diver, we go down to a depth about 70 or 80 feet, and you'll see just a field. I call it like a field, similar to that picture where you just see basket stars popping up all over the place, all different sizes. And they can be almost 3 feet wide and from fingertip, or I guess arm tip to arm tip. And it's just incredible to see that in the wild. And for me, that is one of my most favorite parts of my job, is seeing our counterpart, the wild counterparts of the animals that we take care of at the aquarium. And to me, the Slant Rock is one of my most favorite dive sites.
Amazing. Yeah. And I know we have a really great partnership with the macaw who steward those waters, and that's been a long time partnership, and we're lucky to get to go out there and see the life there. So we are going to bring all of our speakers back onto the Lightning Talk stage for some Q&A. I've seen a lot of really great questions in the chat, but first I want to give you all, our wonderful speakers, a chance if you have any burning questions for anyone here.
So I can go first. I have a question for Sarah. Have you guys seen any problems in getting people to kind of develop that love for spineless creatures?
Yeah, definitely. There's so many factors that are kind of working against them, right? Like, not only are they kind of low or appear low on those characteristics, right? Like, it's hard to tell what parts of their bodies are what. It's hard to tell if they're kind of healthy and thriving, right? Many people, right, don't know what that looks like. And then there's also just been, kind of perpetuated assumptions or misconceptions about those animals. I know, for example, I didn't grow up near the ocean. I grew up in the mountains, and I didn't know that barnacles were animals. I thought that those kind of the shells that were left on rocks or pilings were maybe abandoned shells, or maybe they were incrustations of salt. And it wasn't until someone at the Seattle Aquarium kind of told me the story of, barnacles start their lives as plankton, and then they glue their heads to a safe rock, and then they clamp themselves closed in the dry times. And then when they're submerged in water, their beautiful feet with these like comb-like structures come out to feed. So I think that unless you have someone who maybe is kind of guiding you or helping you to see these animals in a new way, it can be really easy to think of them as objects, and not living creatures.
There were a couple of questions in the chat that kind of struck me as coming from an empathy curious lens that I would love to pose. So one question was about whether crabs, Kate, if you know, do crabs bond with other crabs or any other creatures?
I don't know. It would be how you kind of attach a definition to bond. Crabs are unique because a lot of species have things that grow on them, or decorator crabs will remove things from their environment and place it on themselves to blend in. Like decorator crabs will grab anemones and put them on. Hermit crab shells a lot of times have barnacles growing on them, or the crabs themselves will have something growing on their carapace. So it's not like a friendship bond, but there's definitely things that benefit both creatures that happen. So there's that kind of bond.
Yeah. And maybe a literal bond if they're kind of glued together.
Yeah.
One other question, Luan, that came up was whether nudibranchs are sensitive to light. So if you were to use a flashlight to try and spot nudibranchs, if that would bother them.
I'm sure some species would have a preference towards or away from light, but in my experience of trying to look for them, it's more dependent on what the light is doing with their food source. So it would be more like if there was a lot of light causing there to be a lot of algae, that might impact them. But in terms of me like shining a flashlight on them, I've not noticed a reaction. They don't seem to move differently or notice it. Often I'll go like tide pooling at night and you just have a headlamp, and lots of the other tide pool creatures might react to that, but nudibranchs don't seem to have any sort of reaction one way or another.
One other question, Luann, for you about nudibranchs. Someone noted that a lot of them have wild colors, and they also have some pretty great names that you shared. But they're curious if there's a reason for those wild colors, if those help keep them safe, or if there's another purpose.
So they do. It varies from species to species. Some nudibranchs have colors that help them blend in, that make them difficult to find on a surface, but others have like bright, flashy colors, more as like a warning, like a "don't eat me, stay away from me" kind of situation. But it just varies depending on the species.
So interesting. All right, a couple of questions for you, Bree, about basket star arms. So how do they grow, and how do they use them to get food to their mouths? Do they move the whole arm, or do they move the food down the arm?
Yeah, for the moving the food, I definitely know that answer. So as I was describing that curling of the branchlets and the tiny hooks on those branchlets. So thinking of a tree, you have like, well, I guess thinking of a basket star, they have the central disc, the branching arms, and at that very end, those hooks are actually holding on to the food, and then they are curling that arm all the way into the central disc. They actually put part of their arm, the branchlets, into the mouth, and then take in the food and then remove their arm. And so they're doing that all throughout the day in that current, taking the food from the current in the wild and bringing it to their mouth. So they could be fully open or all the way closed, but usually it's just one arm coming in at a time. And then for the other question, I'm not exactly sure the mechanism in which the basket star's arms regenerate, but I do know that it takes quite a long time. And so that's all the more reason why we need to catch any sort of stress response if we do start to see arms or limbs falling off of the animal. So that's when we do reach out to our vet staff to consult with them on a plan to care for the animal. And I do really appreciate having a full vet staff here at the aquarium as well to rely on for those type of questions.
Wonderful. Well, let's talk a little bit about the important role these animals play in the ecosystem. So Kate, you mentioned some things about crabs cleaning up some detritus, I think was one of the roles, but could each of you think of, especially those who have talked about a specific type of animal, something important that they do in the ecosystem that you haven't already shared? Feel free to jump in when you're ready. Drew, are you ready?
Sure. I'm happy to talk about sunflower stars because they've become really important. We know they're major predators. They're considered keystone predators. And when they were removed by the disease epidemic 21 years ago, sea urchins exploded. And that's because sunflower stars love to eat sea urchins and can mow them down in vast numbers. And so when you take the sunflower stars away, the urchins exploded. Well, that seems great. We like urchins, right? Except urchins really like algae. And so the urchins have basically mowed down a lot of our kelp beds as a release from predation. So I would say sort of the trophic role of a lot of these invertebrates is pretty important, even though sometimes we don't know until they're gone.
Mm-hmm. How about you, Bree? Oh, sorry.
I'm trying to think what else basket stars do in the environment. They are such deep dwelling animals that there is, up until the last handful of years, very few folks have actually worked with basket stars. And so there's really a lot that needs to be learned about basket stars in general, and especially in the wild. So I really don't have a great answer off the top of my head. Kate, can you think of anything? Because since you also work with on our team.
I would maybe talk about, because they kind of filter larger animals or pieces out of the water column. So if there was stuff floating in the water, they're probably grabbing it and processing it.
Yeah.
So.
Well, and
Yeah,
maybe
exactly.
the fact that there's still some mystery also gets to Drew's point that they probably play other important roles that we may not know about yet, but that doesn't mean they don't. And we want to make sure they stick around so that we can learn more and have an intact ecosystem. Luann, what would you like to share about nudibranchs and their kind of how they interact with the broader ecosystem?
I like to think of them as like very specialist gardeners. Now, nudibranchs are they're all predators, but they prey on things that can appear to be plant-like, like hydroids or sponges, things that aren't really moving. And each species will have usually a pretty specialized diet, so they're able to kind of maintain that balance in the ecosystem and keep all those different populations in check. So I just think of them as like a little gardener, like tending to their plot sometimes.
That's a great one. Kate, did you want to add anything about crabs?
I kind of feel like I touched on it all already.
Okay. Well, Sarah, I feel like I want to give you some kind of challenge of helping us build empathy for one of these creatures. Did anything jump out at you in the talks that you would point to as a good starting point for talking to maybe kids about building a connection?
That's a great question. I was thinking about a couple folks were talking about nudibranchs and about their colors and those sometimes those bright colors, and just thinking about a little bit of perspective taking about, obviously a nudibranch isn't making a choice to have its skin, appear a certain color, just like we aren't, but why that might be helpful to them and what threats they might face. So like the more that we can sort of inhabit the worldview of another animal, it kind of helps to make that animal, come alive, and we can start to see them as as an individual and not just a generic a specimen. And that's the thing that I think is one of the hardest things to do in zoos and aquariums, even because we actually are kind of we can perpetrate some of those same kind of language things where I saw an aquarium website that talked about like, come visit our colorful coral reef and the animals that live there. I'm like, oh, but the coral are also animals, right? And so just even thinking about those like really small choices in our language can really powerfully influence the way that people see these animals.
Mm-hmm.
I'm curious. Go ahead, Drew, go ahead.
I just wanted to add something because I loved the notion of how we develop more empathy for these animals. And we've seen it at Friday Harbor Labs in our captive breeding program for sunflower stars. Dr. Jason Hodine is the brilliant one. And we have males and female stars, then we need to breed them. So we need to know which one's which. And so he started naming them. And now when people come to see them, as soon as they find out they have names and that they have distinctive personalities, like Prince is always the one that comes running up to the edge of the tank. Sunny's the one that hangs back a little bit, but she eats a lot more. Suddenly people like look at them like, oh, like they're individuals. And so there's something about the individuality that's important.
Drew, I love that you brought that up. And I had a colleague who would often say that when we give someone a name, we give them a narrative. And, you were talking earlier, I think was it Bree who was talking about this idea of like, yeah, you're in the middle of their story. And we sometimes think about names as being inappropriate to use with wild animals, but names are not uniquely human conventions, right? There are numerous examples in the animal world of animals having distinct calls or distinct markers or identifiers to identify other individuals. And so it's not anti-scientific to designate, some kind of label or nomenclature for a specific individual, and to be able to talk about the patterns or the tendencies that characterize their ways of interacting with the world. I think there's a lot of pushback that somehow empathy and that kind of critical anthropomorphism is anti-scientific, but it's really not. Like the bigger danger is just assuming that the human experience is the only kind of sentient experience in the world.
Well, I'm wondering here in the aquarium, Bree and Kate, where we have, a certain number of basket stars and crabs, how does that individual identity factor in? Do some of them have just good distinguishing characteristics that you note, or how do you think about that in your work with them?
I mean, we don't necessarily name our individual invertebrates. The only ones that we name are our giant Pacific octopus that go on exhibit. But I feel like a lot of our invertebrates, as caretakers, we are able to tell them apart. Either it's from their colors or the patterns that they have. It kind of just depends on the individual invertebrates. But unless it's something that looks overly the same, like maybe some anemones, but even those have their own individual color morphs or color patterns.
Yeah, I totally agree, Kate, with what you're saying. A couple of species come to mind. Well, one thing about the basket stars that's really cool, and I didn't touch on this, is they do have some unique coloration. So in that central disc in the center of the basket star, there is a slight difference in the modeling of the beige to tan to slightly whitish coloration. So you can see specificity between individuals. And if you were to line up a bunch of basket stars in a row, or maybe you go back to the same dive site year after year, you could potentially take photographs. You could potentially see the same individuals and be able to identify them with the markings on their central disc. I haven't been able to work with multiple basket stars in one habitat where that became a critical piece of my day-to-day work. I would say also another invertebrate that is one of my absolute favorites is the abalone, the pinto abalone. And Kate and I both work on the abalone team here at the aquarium. And with our adult abalone individuals, I personally have cared for those in Puget Sound fish, and some of them are now over on Pier 59. But they show little personality traits, and Kate has worked with those individuals as well. And I find that this one likes to hang out on that rock, or this one always rears up on the back of its shell and wants to get its kelp. So I think those are the another charismatic invertebrate that we care for. Of course, the giant Pacific octopus, after several years of taking care of octopus, I can tell you there's different personalities and inklings that one may have over another. And the more I work with invertebrates, I have a greater appreciation and love for all of those little unique characteristics. I considered myself more of a fish person when I first got into this field, and very much over the last 10 years, I've become more and more of an invertebrate person. So
wonderful. Well, it's almost time to wrap up, so I'm going to ask one last question to each of you. If there are people here in the region or elsewhere who want to go out somewhere to a beach or a dock or diving to try and find an invertebrate, so can you tell us what invertebrate you hope they'll look for and what they could do to protect it, whether that's treating that specific animal that they find with empathy and care, or something for their species? So what to look for and what they can do to help. Does anyone want to go first?
I can start. I guess one thing I love about our temperate waters here is I actually consider us kind of unique that we have so many invertebrates that are always out and about. Tropical waters always gets the love for the corals, but a lot of their invertebrates only come out at night, or they're very small and they hide a lot. So I always enjoy diving here because our crabs are always out. They're always out where you can easily find them. And I think just respecting their space and being aware of, if you're walking on the beach, being aware of where you walk, you might not be able to see them, but they're there. Or diving, just being aware of to respect the animals while you're spending time around them.
Yeah, I have another thing to go off of what Kate said is my spark animal as a young child was a shore crab, and it was from walking on the beach at low tide out in South Puget Sound, and I would lift up a rock and look at the underside, and I'd see all the little shore crabs run around, and my grandpa actually would tell me, he's like, now remember, put the rock back where you found it, and that's the home for this animal. And so at a very young age, I started to learn, and those building blocks towards empathy really allowed me to think about, oh, this is the home, I should put it back. And that is one of my favorite things I like to tell people who are just asking, what are the tips and tricks for beach combing or checking out animals in their natural habitat?
That's a great one. Rue, do you want to share
I
something?
would just say one of my favorites would be sea stars. Do you know there are over 80 species in our waters if you look at Kozlov's Key? I mean, the diversity is spectacular. Yes, they're diminished from sea star wasting, but some of the species are doing really, really well. And I guess, look for the ochre star because you see them in a lot of our habitats. And, just don't pick them up. Leave them in their nice shady place where they're happy. I guess if you find them high on the beach, you could move them down. But yeah, they're just so fun to watch them eating mussel.
What about you, Luan?
So, of course, I want people to go see nudibranchs, and I would send them, if you're going for your first time, hop a ferry over to Bremerton Marina, because you will see not only nudibranchs, but so many anemones and feather duster worms. It's just like a whole open aquarium there. It's amazing. And the best thing that you can do to protect these creatures is to tell somebody else about it. Like if somebody's walking by, be like, did you see this cool thing down here? And then that way you can impact how people think about their world, which is probably the most important thing we can do.
That's a great tip. Sarah.
Yeah, I would echo that from Luan, but just this idea that these are living animals, because that is new information for so many people, that these are living animals, and they're sensitive. They're incredibly sensitive to touch and the kind of tactile conditions. And so if you do decide to touch a sea star or a sea cucumber, to really gently, one wet finger with about the same amount of pressure that you could put on your earlobe without bending it, is the right amount to keep that animal safe and healthy.
Well, thank you all so much. There are lots more questions in the chat. Everyone was very engaged, and we will find other ways for you to get answers to those questions. But I want to thank everyone for joining us tonight. Please put some thanks and kudos in the chat for our wonderful speakers. And if you would like to check out past episodes of Lightning Talks, you can visit our Lightning Talks page on the Seattle Aquarium website. We have great ones on topics from octopuses to salmon to ocean ancients. And we have another one scheduled that you can tune in for on July 17th. We're going to be talking about seahorses and sea dragons. So don't miss that. And if you have enjoyed this free science event tonight, please consider a donation to the Seattle Aquarium to help keep these events free and accessible for all. And with that, I'll say good night and see you all next time.
Bye, everybody.
Good night.
Bye-bye.